The biologist I chose for my role model project is Kristen Marhaver. Kristen first became interested in coral reefs when she first went scuba diving at 15 years old. Her father was in a scientific career based on cancer research, so she grew up around science. At university, she studied biology, but mainly courses that were about cell biography and cancer biology. She received a Bachelor of Science in Applied Biology from the Georgia Institute of Technology. She also received a PhD in Marine Biology from Center for Marine Biodiversity and Conservation at Scripps Institution of Oceanography. However, Kristen knew she wanted to study marine biology and hasn't looked back since. Kristen's main hobbies are photography, painting, dancing, and tropical gardening. She enjoys doing these activities when she isn't in the water. The thing I admire most about her is that she is a successful woman and that has always known what she wanted to be since she was younger. I relate to her because I've always wanted to be a pharmacist since I was a young girl, so she gives me hope since she followed in her dream. I identify with her because her father was involved in cancer research. Sadly, two of my aunts died from cancer, and my grandma is currently suffering breast cancer. So her father's job really stood out to me. Kristen is involved in marine biology, especially coral biology. She studies the ecology, behavior, and reproduction of coral reefs. The main questions she is focused on are how do corals reproduce and how do we keep young corals from aspects that are constricting their growth. She is even now developing bacterial tools to improve coral survival at all life stages. The most recent paper Kristen was authored on was Inheritance of Somatic Mutations by Animal Offspring. This data figure I have shown from the paper as well. The experiment performed was that the scientists sampled 19,696 single nucleotide polymorphism loci with a microarray to determine whether the neighboring colonies represented different genets. The parent colony and all five of the neighboring colonies belonged to the same implemented genet. The main conclusion for this figure is that the average pairwise genetic between samples were extremely low, both among 10 parent colony samples and among all 6 sample colonies. This data therefore eliminated both types of outcrossing as likely explanations for spontaneous development of the eggs. Furthermore, the low pairwise genetic distances measured within and between colonies pointed to the existence of somatic mutations in the parent coral and neighboring gametes of the same genet, which could possibly be identified in the resulting offspring. Kristen has discovered a lot of amazing stuff so far. A couple of things are discovering the spawning time of threatened pillar corals and raising the first lab-reared juveniles. Also, she discovered bacteria that was a Q coral settlement. Lastly, she observes how quickly and healthy corals can regrow. I am very happy who I choose to be my role model biologist.